So we're going to talk about science in single molecules, how industrial basic research at low temperatures 30 years ago led to super resolution microscopy and beyond. So this is a very multidisciplinary topic, and we have a very broad audience. So I bet that there may be something of interest to almost everyone here. So if you will go along with me as we move forward, I'll tell you quickly how the talk will be organized. It's really a story of physical chemistry, but also ultrasensitive detection and industrial basic research that has unexpected effects spanning physics, chemistry, engineering, and biology. So this will give me a chance to talk about the early days of single molecules, how they were detected in the first place, how that became super resolution microscopy with single molecules, and then I'll talk about how you can use this technique in cells to learn a great deal about the nanoscale inside these wonderful machines, as well as molecules in solution. And finally, I'll talk about energy harvesting molecules, molecules that are part of the photosynthetic system. So it's a broad range. Let's go back to 1985, the middle of the 1980s, before single molecules were first detected. And the air was filled with can they be detected? And it wasn't so clear that it was possible. In fact, there was this really interesting statement by this great founder of quantum mechanics, Erwin Schrödinger. He said in 1952 that we never experiment with one electron or one atom or one molecule. And if we did so, that might be something completely ridiculous. Maybe, for example, similar to building a dinosaur and having a dinosaur be in the zoo. But this is a wonderful and fascinating statement from, of course, a great scientist. But it will give me an opportunity to sort of explain. You see, people thought it was impossible to detect a single molecule. So one of the key ideas is to first realize how we became to believe that it was possible. Of course, this gives me a quickly opportunity to provide a thought for you to remember from this part of the talk, a signpost. Beware when Nobel laureates say something cannot be done. So that actually provides impetus for scientists to prove the person wrong. And you heard that earlier in Professor Kip Thorne's speech as well. So that's the idea. How do we believe that it's possible? So the early steps toward the single molecule detection were done when I was at IBM Research in San Jose, California. Now, how is that possible? Well, we were interested in storing data, storing digital data in the color of the absorption spectrum of molecules. So using, at low temperatures, the ability of tunable lasers to an inhomogeneously broadened spectral lines, you could tune the laser and at different colors produce a bit that's a 1 or a 0. So this would allow 1,000 bits to be produced in any one spot inside the sample, something called frequency domain optical storage. And so it was an interesting idea, an interesting sort of technological thought to be able to try to make this work. But the key point is that at this industrial research lab, in one of the great ones of the 50s to the 90s, we could not only explore the new technologies, but also measure fundamental properties and fundamental science underlying the idea. So that's where we start our story. The key idea was we were able to ask a question beyond the frontier of knowledge at that time. Here's the question. This is an optical absorption line at low temperatures of the molecules pentacene, which is not showing up for you, and paraterphenyl for some reason. So let's see, I will try to restart so that we can get that molecule to show up. I'm not sure why it's not showing up, but let's try it again. So the idea is that pentacene, which is an absorbing molecule in the visible, has an optical absorption band that looks like this. Sorry about that. And so the question was, this is the absorption of many molecules, many thousands of molecules. It's pretty narrow because it's low temperatures. But the question was, is this spectrum completely smooth? Because the bits that we are going to write are going to be written in this absorption line. So we have to be able to ask the question, well, what's the background? Is it smooth or not? And no one had answered this question, but in 1987, my postdoc and I measured the structure on the top of this absorption line, and it looks like this. It's amazing. It's quite a fascinating spectral structure, and this is not noise. This is something that repeats. If you measure it once and then measure it again, you see the same features. It's coming from a statistical effect. The amplitude of this is the square root of the number of molecules in resonance. So it's a bit of statistics, it's a bit of spectroscopy, it's a background noise signal that might influence this particular technology. But nevertheless, we discovered this and showed that it was there, and this led us really to single molecules. Because once you see a spectral feature that scales as the square root of n, then you know it's only the square root of n times harder to go to the single molecule limit. So in 1989, we did that. We went to the single molecule limit for penicillin and paraterphenyl, and the single molecule regime was first breached in these experiments where we measured the optical absorption of one single molecule. It was done by FM spectroscopy, a beautiful and very powerful high-resolution spectroscopy technique. And the shape of a single molecule should look like a W, and you can see that W here in the spectrum. And so this opened up a new area of science, the fact that we could see a single molecule with light. A year after these original experiments, another measurement of the optical absorption of the single molecule was done by Michel Horry, where he measured the emission light, the fluorescence coming from these molecules, one at a time. And so that's another way to measure absorption, but it's more useful in terms of signal to noise. So everyone switched to fluorescence excitation. Now, so we detect a single molecule. But the point that you might be asking is, well, why? Who cares? Why? What good is one molecule at a time? And so let me give you an analogy to show you why this is an interesting experiment. It's a baseball analogy, and you might use this to explain it to your students if you're a teacher or an educator. Here's the idea. Well, back in 2004, the World Series of Baseball was won by the Boston Red Sox, and one of my students was a Red Sox fan, so she made this slide for me. This shows the performance of all the teams playing that year in the finals, and you can see that there are listed here team batting averages. So these are the batting averages averaged over the ensemble, that is averaged over all the players on each team. Well, we know that you also have data on the individual players in the team, and you can make a distribution like this that shows the different batting averages on this axis and the number on this axis. And so you can see what we call the probability distribution of this measurement, and you see something strange over here. There's a bunch of players that have very, very low batting averages that you might not see if you only had the average itself from the whole team. Well, these, of course, are the pitchers. They can throw the ball pretty well, but they don't hit it very well. So by being able to measure the behavior molecule by molecule, in this case, it's player by player, you can measure this distribution and get greater insight into the problem. So we essentially are translating this idea from baseball to molecules. And you can ask the question, are the molecules the same or are they marching to different drummers? So this idea of being able to remove ensemble averaging is very powerful. That's what happens when you can measure one molecule at a time. You can explore heterogeneity and determine whether there are some differences that you didn't know about. You can also watch each molecule as it changes in time if that happens. Watch the dynamics of individual molecules. That's something that would be smeared out if you averaged over many billions of molecules, and so on. We also can use these measurements to look at local environments. Since each molecule is about a nanometer in size, you can measure and detect and sense nanometer scale interactions. And the fact that the molecules are very small probes will lead to super resolution imaging, which I'll talk about in a moment. But this regime of single molecules can also be used for sequencing DNA. And that's happening with several companies right now. So signpost number 2, even in this industrial research environment, when the exploration of a technology includes a will or a willingness to explore fundamental basic science, great discoveries can occur. This also applies, of course, to the university. As long as you have an open mind to fundamental basic science, you might discover things that you didn't expect. Well, there actually were a lot of surprises. Because this new field had not been explored, let's talk about the surprises. So here I'm showing you a bunch of spectra of a single molecule. So the horizontal axis is frequency, and this is really a spectrum of the molecule. But you see something strange going on. The sample is at 2 degrees Kelvin. It's a crystal, and there should not be any dynamics. yet you see the molecule is moving from one frequency to another. Its resonance frequency is changing. And this surprised us. We didn't expect it in this crystalline system, and it turns out it's coming from low temperature dynamics in the crystal. But if you imagine, there's another way to think about this. If I just fix my laser frequency at one color here, then what you observe in time is that the molecule is in resonance, out of resonance, in resonance, out of resonance, in a fascinating blinking pattern. We call it blinking, which is quite random. And you might think, well, they're blinking, and so they're not very stable. But we have an application for this idea later on in the lecture that I'll tell you about. Another experiment that was done at that time was to control the molecule with light. Here's a single molecule, a spectrum of a single molecule. It seems to be stable as we measure it multiple times. But then if you bring the laser into resonance with the molecule will disappear, appear to go somewhere else in frequency space, far off the screen. Okay, you've driven it away. Maybe you photo bleached it like you photo bleach your blue jeans. But in fact, if you wait another moment, this molecule comes back to exactly the same frequency, and you can drive it away again. So reversibly controlling the molecules with light was another feature observed at room temperatures in these very early experiments. Okay, so low temperature is exciting for basic science, but we knew that if you could go to room temperature, the applications of this idea would broaden dramatically. So in the mid-90s, a number of people, including us, went to room temperatures for the observation and detection of single molecules. We're still pumping molecules from a ground state to an excited state with a laser, and we're collecting the emitted fluorescence. And we're trying to cycle through this many times to collect a lot of photons. At room temperature, the molecules that we use are these rhodamine dyes or other fluorescent labels that can be attached to proteins. So here's, for example, or oligonucleotides. The label is attached to the object of interest. The label can even be a fluorescent protein like GFP. Now, at room temperature, the way you want to think about the experiments is imagine focusing the laser down to the smallest possible spot. It won't be infinitely small. In fact, it will have a width that's limited by diffraction to roughly lambda, the wavelength, divided by 2 times the numerical aperture, or a few hundred nanometers. But remember, the molecules themselves are just a couple of nanometers in size. So in order to pump only one at a time, we have to dilute the molecules. We have to spread them out in the sample to see single molecules. But you can do that, and then the light that you collect, if it's strong enough, will be the light from a single molecule. Well, now what you can do with this is to image immediately at room temperature molecules of interest. Here's an example from biology. A particular protein called the major histocompatibility complex of type 2, MHC2 proteins. They bind an antigen, a small peptide, and we put a fluorophore, a label, a Psi5 label on that antigen. So that when you imagine this object in the cell membrane, when we pump with light, we collect the fluorescence from this label on each one of these MHC2 proteins. So here's a picture of the light coming from a cell, a living cell, the edges of the cell, and each of these little spots is the light from a single molecule. And I want to emphasize that these experiments were done with a good microscope, a high-quality microscope with a very high sensitivity detector. But you can even detect the light from the single molecule with your own eyes. Looking into the microscope, when your eyes are dark adapted, you can observe these little pinpoints of light yourself. That's, of course, shows you how sensitive the human eye is, but shows you also how bright these single molecules emitters are. More interesting than just one image, of course, is the movie of how it changes with time. And when you run the movie, you see there's this amazing dance of single molecules on the surface of the cell. And this is occurring in your cells in your body right now as we sit here. The molecules move around due to thermal excitation and Brownian motion. Moreover, you can see other things happening here. You can see not only do they move, which you can study scientifically, you also see them turning off. You see them present and then turning off after some time period, or maybe even turning back on again. So that's normally the photobleaching process. And since the molecule turns off in one step, that's proof that this is a single molecule that was being observed. This room temperature regime, of course, exploded into many interesting experiments by many groups all over the world in those days. So we were exploring different kinds of single molecules at room temperature and happened upon another surprise. Okay, remember, new regimes, new surprises can occur. We decided to look at individual yellow fluorescent proteins because fluorescent proteins were being used by many people to label specific structures inside cells. You can let the cell make the label itself. So a yellow fluorescent protein, no fluorescent proteins had been observed at the single molecule limit, so we set out to do that, and we saw these beautiful single molecule images as well. But again, surprises appeared. For this fluorescent protein, it turns out to blink at room temperature. That is, the molecule would emit in successive frames of a camera, and then turn off for some time period, but then turn back on again, and off, and on again, and off, and in a random pattern, like I was talking about before. This is a signature that in the fluorescent protein, we can pump and collect photons for a while, but after some time period, the molecule goes into, the protein goes into a dark state that represents some kind of photochemical or photophysical change in the local environment of the molecule. But it can return to the bright state and start emitting again. And so that was what we would call blinking, on and off blinking. We also found that the molecules would go into a deep dark state that does not recover for a very long time unless you use a little bit of blue light to turn the molecule back on. Optically controlled emission was possible then for fluorescent proteins, a used up molecule, a restored molecule, a used up molecule, a restored molecule. So just like at low temperatures, blinking and photo control were present. And this was thought to be important for applications like optical storage again. Many people worked on much more optimized switchable fluorescent proteins. But signpost number 3 now, when exploring the new regime, then I've tried to illustrate to you with my own work that we cannot predict in advance some of the wonderful surprises that may occur. Well, in addition, these surprises can lead to unexpected and unplanned applications. So that's the interesting part about this type of fundamental basic research. So now I want to talk about the Nobel Prize and how what I've told you so far leads to super resolution imaging. But before I do that, I want to ask you a question, okay, that may surprise you. What is the next best thing to being picked for a Nobel Prize? Okay? You might ask yourself, well, for me, it's marrying my wife, having our son, and so on. Those are the next best things than a Nobel Prize. But I want to give you kind of a different answer to this question. The answer is being picked by Milhouse on The Simpsons. So believe it or not, in 2010, okay, this is 4 years before the Nobel Prize, there was a particular episode of The Simpsons TV show. You can look it up on the web if you want to see it. And what's happening? With families waking up in the middle of the night, okay, like it is for me right now, the middle of the night, and they wanted to know, why do I have to get up so early? What's going on? Why do we have to go watch the TV? But then Homer Simpson says, well, we have to watch the Nobel Prize announcements. So they were all getting on the couch and they're ready to watch the TV, and it turns out they were having a betting pool. They were betting who was going to win the Nobel Prize. And here are the different prizes. And here are the different family members, and this was shown on the TV screen. And then right here in the middle, who do you see? That's my name, if you can't read it from the audience, on the TV. So, like I say, this is the next best thing to being picked for the Nobel Prize, to be on The Simpsons, right? Well, they're very impressive there on the Simpsons team because they also predicted another prize winner. Here's Ben Feringa, who won the Nobel Prize in Chemistry just this past fall. So anyway, let's go back to the real thing. And to talk a little bit more about the importance of the Nobel Prize, let me just remind you of the grand challenge. What are we really trying to achieve with all of this research? We would like to observe the nanoscale. If you're thinking about cells, we want to observe the molecular machinery that's at work inside living cells or complex systems. So here's a bacterium. It's a pretty small object, only a few microns in length, a few 100 nanometers across, 500 nanometers across. And if you blow up the image a little bit more, there are fascinating and important, interesting small proteins and machines like the ribosome inside this cell. Here's a 20 nanometer scale bar. These objects are quite small, down in the nanometer size scale. You would like to be able to image these objects and image their structures. And so you need high sensitivity, high specificity method. You need the ability to image living cells, and you want very high spatial resolution. And you might be thinking, well, why don't we use electron microscopy or some of these other methods that have very high spatial resolution? Unfortunately, they don't work in living cells, and they don't have the specificity of fluorescence. Remember that fluorescent label can be attached to just one kind of protein, so that the signal we get comes from just that protein and not from all the others. The only problem is, remember I talked to you about the size of the laser spot. On the scale of this bacterium, the smallest possible laser spot with visible wavelengths is too large to properly image all of these objects of interest. So that's the problem. Since the beginning of microscopy, optical imaging has had too low of a resolution. So the prize was given for super resolution fluorescence microscopy, and the other prize winners are shown here. Stefan Hell used a method called stimulated emission depletion. Eric Batzig and I worked on the single molecule approach to this. So I'm only going to talk about the single molecule approach for the rest of the talk. Okay, so the idea is you want to circumvent the optical diffraction limit. And let me explain how this works, or the key idea, slightly differently with this picture. This is a bacterium, again, where we've labeled a specific structure. Is there something over there that's smoking a little bit? I wonder what's going on. So there definitely is something smoking. Pardon? Oh, that's a mosquito fogging. Okay, thank you. Good? Glad to hear it. So this bacterium has fluorescently labeled proteins inside it, and you might ask, I want to see their structure, so let's just go buy the most expensive microscope we can find, then we'll see the structure. So here's the most expensive microscope you could buy in those days before the super resolution, and you don't see the single molecules. You just see a very blurry, fuzzy picture. Now, why is this? Remember that the labels we're using are very small, only a few nanometers in size. But the problem is occurring because the optical image of this near point source appears to be large, a few hundred nanometers in size. This is due to optical diffraction, to the Abbe diffraction limit, and Ernst Abbe in the late 1800s realized this. Lambda over 2 times the numerical aperture is the resolution limit. So in the visible, it's about half the wavelength, so it's about 200, 250 nanometers. So that's the problem with optical and fluorescence microscopy, but super resolution changes this image into this image. A tremendous improvement. In fact, a factor of 5 beyond the diffraction limit I'm showing you here. So the incredible level of additional detail is stupendous. It's not just a small effect of only 15%, 20%, 50%, or whatever. A factor of 5 increase in resolution allows you to see a great deal more. So how does this work? How does the whole idea accomplish this super resolution feat? Well, there are 2 essential ingredients needed to make the idea work. First of all, we have to super localize single molecules. What does that mean? Well, by analogy, let me just ask you to think for a moment about this beautiful little mountain in the middle of a lake. It's called the Cinder Cone. This particular mountain is in Crater Lake, Oregon. It's very beautiful. Go see it if you can. I have my obligatory scale bar here, 120 times 10 to the 9th nanometers. Well, you know that if you simply take your cell phone and walk up to the top of this mountain, you can read off the GPS coordinates of the position of the mountain, okay? That's very similar to what we're doing with single molecules. Here is an image of a single molecule in a bacterium. I'm showing the image now in 3 dimensions, where the colors and the z-axis is just the brightness of the object, the brightness of the molecule. You can see that the molecule is broad. That's the diffraction limit. It's a few hundred nanometers in width. But we can use a fitting algorithm. We can use the measurement of the shape of this mountain to determine the position of the peak to precision that's much better than the diffraction limit. Put that another way, the image of this single molecule, if it's spread over multiple pixels, the different pixels sample the shape of the image. And so those measurements in the pixels can be used in a fitting function, for example, a Gaussian function, to determine the position of the molecule. And this determination has a precision that depends upon 1 over the square root of the number of photons detected. For example, if you detected 10,000 photons, then you could go 100 times below the diffraction limit in terms of localizing the position of the molecule. So that's super localization. It's not really a new idea in science. It's been used in many fields, but here we're doing with a nanoscale emitter, a single molecule in a cell. It's a very powerful idea, a powerful measurement. Okay, but it doesn't give us resolution. This by itself doesn't give you a way of separating two molecules that are very close together. And that's what you need for resolution, all right? This only works if the molecules are separated from each other by a large amount. So to solve that problem and really achieve resolution improvement, what we do is to simply add the second technique, which is called active control of the emitting concentration. So how does that work? Well, we use the fact that many of these emitters are going to have the ability to turn on and off. That is, they can be blinking molecules, or they can be molecules that we can use with optical control, or that we can use chemistry or other methods to control the number of molecules that are emitting at any given time. And so how does that help us? Well, here's a structure we would like to observe. And so if you let all those molecules, you see the many molecules labeling the structure, if we let all of them emit at the same time, then we see the blurry image, because each molecule produces a fuzzy ball on the detector. So to solve the problem, then we use this control mechanism that I'm now showing you here, that these molecules that you want to use for this method must have a dark state and a bright state. And you can control the fraction of time you're in the bright state or the dark state. So then you use that control mechanism to only turn on a few molecules at a time. And you measure their positions by fitting, and then, for example, photobleach them. And then you do it again. You turn on another subset of molecules, find their positions. All of these positions are recorded in the computer. They're all saved. And in the end, you have a long list of positions. And you use the computer to reconstruct those positions. And this gives you detail beyond the optical diffraction limit. So nowhere in all of this have we broken any physical laws. We're simply using the fact that in the time domain, having different molecules on at different times, you're able to use them to sample the structure with high precision. So this beautiful idea I first heard about from Eric Betzig in April 2006, and he called it PALM. Then Xiaowei Zhuang called it STORM, and Sam Hess in the same year, in the same month pretty much, FPALM. Then many other people created some new acronyms for the idea: PAINT, GSDIM, BLINK, SOFI, etc., DSTORM. All of these acronyms are pretty much talking about different mechanisms for achieving this active control. We used that yellow fluorescent protein reactivation that I told you about earlier. And so, but we didn't add an acronym to our particular paper, so that's obviously a mistake, because if you don't have the acronym, then the paper can be forgotten. But nevertheless, to rectify that problem, here, let me give you another acronym that's mechanism independent single molecule active control microscopy, which I hope explains to you the idea. Fine. So this particular method of super resolution then has been applied to many, many different systems. Bacteria, cells, and even polymers and other samples. But let me illustrate it directly here with some real data, some raw data. Here's the first image, okay, of a sample of a cell where the microtubules have been labeled with a molecule that will blink very strongly. So imagine that the tubulin molecules that make up microtubule have been labeled with an antibody which carries a small fluorescent label that will blink quite a great deal. So in this first frame of the movie, then we localize all the molecules, and there are white spots on the right side that you can't quite see, but they're very tiny. In the next frame of the movie, other molecules turn on. Remember, it's blinking very strongly. The next frame, more molecules. And then ultimately, if you just run the movie with many frames, then you can see that this fantastic structure appears at high resolution, far beyond this diffraction-limited image that's on the left. This is the super resolution image on the right. So the idea is wonderful, because it can work whenever you have a mechanism that allows you to control the number of molecules that are emitting at a given time. So I'm now at signpost number 4. Super resolution fluorescence microscopy really gives us a new window into the nanoscale because the resolution improvement is not small, and it's broken this fundamental diffraction limit that limited all previous optical microscopy. Currently, people have pushed down to the 1 nanometer resolution in certain cases, but 10 nanometers is more common for most experiments. Moreover, the idea required fundamental scientific advances to put it together. For example, imaging single molecules, as well as the surprises of blinking and switching that were observed when we went to the single molecule regime. I want to give you a few very brief examples of imaging that was done with these super resolution methods, and I cannot be at least a bit comprehensive, but I just want to show you a few. One example is you can now look at the aggregates of proteins that form amyloids and produce certain diseases, for example, Huntington's disease. Huntington's disease is caused by the Huntington protein when it's mutated to have a very long sequence of cues in the middle of the protein. And so you can fluorescently label these mutant Huntington proteins and look inside the cell how the aggregates form. Here's an example. In a neuronal model cell, you can see a very, very large inclusion body that's normally measured in conventional microscopy, but all around the inclusion body are very fine fibrils that are formed from the mutant Huntington protein that would not be observable without super resolution fluorescence. These appear out in the axonal growths. Here's a close-up of one of them. The diffraction-limited image is just a blur, but the super resolution image in reverse contrast gives you a lot more detail. The point here is that this doesn't immediately solve Huntington's disease, but it gives you a way of measuring these key protein aggregates with far higher detail and much, much higher precision, so that you can then see what the size changes occur when different kinds of treatments are applied to the Huntington problem. Another example of a beautiful measurement, some discovery that came from super resolution fluorescence, was done by the Xiaowei Zhuang lab, who looked at the axons of neuronal cells, and specifically actin and spectrin were the proteins that were fluorescently labeled. And what you see here in one of these axons is this amazing periodic structure of actin and spectrin along the long axis of the axon. This was not observed before the super resolution fluorescence microscopy. And a third example brings in another area of science that we spend a lot of time on, doing all of this in 3 dimensions with 3D imaging. It turns out that you can change the microscope. A regular 2-dimensional camera can produce 3-dimensional images if you carefully change the microscope in a fairly simple way to encode the Z information of the motion of the location of the molecules, as well as the X and Y information. And this is an example of applying the 3D methods to proteins in a set of bacterial cells. There is yellow and orange is labeling a particular protein called crescentin inside these bacteria, which makes a helical-like shape. And the surface of these cells has been labeled with a different fluorophore, and it's shown here in white and gray. So 3-dimensional imaging is now routine, but there's a wonderful set of scientific problems that we solve to make these images be as detailed as possible and as precise and also accurate as possible. But I won't go into all the details of the optics today because of time. Signpost number 5, however, super resolution imaging is now providing an amazing new view into the nanoscale structures that are present in cells 5 times, 10 times or so beyond this fundamental diffraction limit. This allows us to have a deeper understanding of cell biology, and that can be done also on materials and polymers and so on. But at the same time, this may lead to new strategies to affect pathogenic processes, such as amyloids or cancer and other types of cellular diseases. So I've told you about some things that you can do with single molecules. But let's summarize a little bit and talk about what other things that can be done with single molecules. First of all, I've talked about how you can super localize a molecule to find its position to accuracy much better than the diffraction limited width, and how to make a super resolution image, which is observing singles, labeling the structure at different times, then putting that together to get the detailed image. But there's something else you can do with these single molecules. By the way, in these cases that I'm talking about, you don't want the molecules to be moving during the imaging process because it takes some time to build up these images. It's a sequential process of sampling the structure. So on the other hand, you want to sometimes see dynamics. You want to see fast motions. You want to see motors moving around. You want to see proteins diffusing inside the cell. So if you want to do that, then you don't do super resolution, but you simply just watch the single molecules move themselves, work at low concentration, and measure the motion of the single molecules. Now that might be something that's due to, as I say, diffusion, but it also might be due to some other process inside the cell where the molecules are moving around, or on the cell surface as well, or inside a polymer, or in other sorts of situations. So this is really recording a trajectory of the single molecule, and you can do this at fairly high speed in the millisecond time regime, and also get very high precision of 10 to 15 nanometers on the localizations of the molecules. I want to give you a quick example of this idea as well. Here's a situation where we applied this tracking, okay, this measuring the motion of single molecules, to explore the motion of chromosomal loci in budding yeast. We're doing this in 3 dimensions using a special optical method called double helix point spread function, DHPSF. I'll tell you very briefly about that. So the experiment here was to label the 2 copies of the GAL locus, the GAL gene locus, in the yeast, and then in 2 different colors. So that in one color we're going to see 1 copy, and in the other color we're going to see the second copy. And then we're imaging these fluorescent labels with this double helix point spread function. The double helix point spread function converts a single spot into 2 spots, and then those 2 spots turn relative to one another, and that is the encoding of the Z position of the molecule. So you can see in these different frames that the 2 spots are beginning to turn, which means that that particular molecule is moving in the Z direction. Here's the other color channel doing something slightly different. So you get this 3-dimensional information for both colors at a frame rate of 10 hertz. This is just showing a simple 2-dimensional projection of those motions. Now, you know, a lot of people have been imaging molecules with, with 2-dimensional imaging, 2-dimensional microscopy, but the 3-dimensional microscopy that we have has far more information. This is showing the motion of one pair, the velocities in each of the pairs of orthogonal directions. So there's all of this beautiful, precise information about the motion of the molecules, which you can use to calculate autocorrelation functions and cross correlations and so on. Here is a plot of the 3-dimensional motion, now using time encoding of these 2 different loci over a fairly long time interval of up to 7 seconds. Sorry, even more than that in this case, up to 30 seconds. So my point here is that these single molecule methods can be applied to measuring motions like this that I'm talking about. Here we're trying to understand why those 2 different copies of the GAL locus are moving in the way that they move. What controls their correlations? Are they packed together inside the chromatin? Are they separated in the chromatin when the gene is turned on? Do they do something different when the gene is turned off? And so on. There's a lot of fascinating questions to be answered inside the nucleus that are now being addressed with these super resolution and with these single molecule tracking technologies. So that's a case of observing motion. I'm just giving you one example, and there are thousands of experiments going on looking at single molecule motions today. I want to talk about one more method before we wind up here. What about single molecules that are in solution? Not in a cell, but in solution. And we're interested in single molecules in solution because in some situations you don't want to perturb a molecule by tying it down to a surface or encapsulating in some other structure. There's some situations where you'd like to just observe one biomolecule and see how it behaves as a function of time. And what we want to do is to do this in a solution. So we have invented a new kind of trap called the Abel trap, which stands for the anti-Brownian electrokinetic trap. That is a machine, a device that suppresses Brownian motion, and then we can measure the brightness, the lifetime, the spectrum, and the polarization of a single molecule, all at the same time and as a function of time. So it's quite a fascinating device that can be used for lots of interesting experiments. So here is, for example, a trapped molecule, trapped in the Abel trap in solution. You saw it emitting for a while there before it finally photobleached. But you are able to observe the molecules typically for many seconds. During that time of many seconds, we can measure multiple parameters. For example, the brightness of the molecule. And we do this because the laser beam is moving in a special pattern in this Abel trap. That time average is a uniform optical excitation. So we have good information about the amplitude of the brightness from the molecule. This was a particular molecule that was labeled with 8 fluorophores, and so you can see it photobleaching in a sequence of 8 steps. That's one parameter, brightness. Another parameter, lifetime, excited state lifetime. We can pump the molecules with a pulsed laser and look at the time delay between the emitted photons and the pumped photons, and then histogram those delays to get the excited state lifetime for the single molecule in the trap. We can also measure the spectrum of the molecule in the trap just by dispersing the emitted light and then recording that on a spectral detector. For example, here's a single molecule in the trap and its optical emission spectrum. So we also can do polarization. The point here is to observe molecules that have interesting photodynamics. And the example I would like to talk about here is to connect all of this work to problems in the energy arena. In photosynthesis, for example, there are many antenna proteins that are involved in absorbing light and then transferring that excitation down to the reaction center where charge is separated. And so we can use our machine to look at individual pigment protein complexes that are photosynthetic antennas. Here's one example. This is the allophycocyanin molecule this particular antenna is naturally fluorescent, so we don't have to label anything. The monomer has 2 chromophores, two fluorescent molecules in it, and the entire structure is normally a trimer. So let's just look at this monomer, this one monomer. Let's trap it in the Abel trap, and here I'm showing a couple of examples of single molecules trapped in the Abel trap. The brightness you see when the molecule comes into the trap fluctuates a little bit, but it has sort of a relatively constant value. But after some time period, it jumps to another level in one step. And that, of course, is always surprising to me. This is a complex system with many, many atoms, many amino acids, and these 2 fluorophores, and yet we see a digital change in the brightness as it changes from one state to another state. So we call these states, and for each state, we measure not only the brightness, but also the polarization, the spectrum, and the excited state lifetime in other cases. Here's another single molecule that has a slightly different trajectory before it photobleaches. So we can make these sorts of measurements for hundreds to thousands of single molecules and try to correlate the changes in brightness, emission, polarization, and so on. Here's an example of that data. So on this plot, what I'm really showing you are many little black spots, and in each black spot, that's really coming from a measurement of a single molecule's brightness, polarization, spectrum, and lifetime. The colors are a Gaussian weighted estimate of the density, and you see that the molecule pretty much starts out when it first comes into the trap as a bright molecule with the highest brightness and a particular polarization and a particular spectrum and a particular lifetime. And these 3 clusters, of course, are the same clusters, but in multiple dimensions. And we believe that's the intact system with the 2 chromophores, alpha and beta, completely intact. Later on, as you go to other states, you see that they correspond to changes which we can interpret as a photobleaching of one of the particular chromophores. Here's when you photobleach the other chromophore. And then finally, there's another state that comes from one chromophore becoming a quencher for the first, where it continues to shorten the lifetime of the first one. So the point here is that by looking at these photodynamics in multiple dimensions, measured multiple parameters, measured molecule by molecule, we can extract this model for the photodynamics and the behavior and measure the coupling between the 2 chromophores. What is the degree of energy transfer? What happens when one photobleaches? And so on. What are the lifetimes and probabilities of transitions between all of these states? So even though these 2 pigments are chemically identical, they give rise to this fascinating, interesting, non-degenerate, non-emissive properties of the molecule. All right, so that's signpost number 6, finally. Beyond super resolution, fluorescence microscopy, which I talked about in some detail, single molecule tracking, and single molecule gives dynamical information down to the millisecond time scale and beyond. And trapping of individual molecules in solution enables dynamical study without surface attachments. So I've given you this sort of broad overview of many aspects of single molecule experiments, but let me try to summarize them in a different way. This area of science not only affects chemistry, but also affects physics, also affects biology. And many different physical processes have now been explored at the single molecule level. And of course, I showed you that super resolution fluorescence is one of the newest things that can be done with these single molecules. All of this progress occurred because working at this ultimate limit has been pursued by a number of researchers all over the world. So now there's a number of students in the audience, and of course there's a lot of teachers and educators. So I want to leave you with a few lessons to tell your students or to tell your children or to tell your family, okay? And so the first lesson I'd like to talk about relates to this medal up here in the corner. This is the Nobel medal, and I know that many of you have seen this picture of Alfred Nobel, which is on the front of the Nobel medal. But I wonder how many of you have seen what's on the back of the Nobel medal. It turns out on the other side of the medal, there's this beautiful sculpture engraved in the metal, and this is it's an inspiring picture, truly inspiring, because here on the left is nature, natura, it says here. And nature is carrying a cornucopia, okay, of riches for the world, food for the world, energy for the world, many things for the world. On the right is scientia, and scientia is lifting the veil of nature. To me, this is a beautiful and incredibly inspiring picture, okay, that can drive us in our work. So I want to say to the students, science is fun, and help lift the veil of nature. That's what you can think of your work as doing. Thank you. But more importantly, there's other things to think about. You need to find your passion. It's truly important to find what really drives you. Don't do something just because someone else told you to do it. You have to want to do it. Science requires this because it can be very hard work. You have to be determined and persistent and methodical. You need this determination to be able to get through the times when things don't work so well. That's part of science. When things fail, it tells you something. It teaches you something. You should use it as a learning experience. Use it as something that will take you into a new arena, because when it failed, it's because you didn't quite understand something, and that's the thing to understand to move forward. Ask how things work. Ask in detail how things work. Well, of course, we do that as scientists all the time. Everyone, all the delegates here do this. But at the same time, we live in a society where lots of people don't ask this question. They simply use their cell phone without thinking of how it truly works. We need people to ask this question deeply, because we have to understand technology, to understand science, to push the world forward. You have to push beyond conventional wisdom and question assumptions so that you can invent and discover new things. Really is an important message, as I've sort of illustrated multiple times. You want to keep your eyes open for surprises. Sometimes when it doesn't behave as you expect, that actually, it could be a mistake, so make sure you're not making a mistake. But if there's no mistake and it's still behaving in a surprising way, that could be the key to a great new discovery and a great new step forward. We all do this, of course, because science provides this rational and predictive way to understand our world. And we need that to maintain our standard of living and to improve it for all the people of the world. So I want to now, in the end, thank my past students, postdocs, collaborators, and the current team. And here's the current students. Well, of course, if you're a spectroscopist, you might understand the joke in this picture. You know, we have some light sources and some gratings on the left, and then that turns into a red, orange, yellow, green, blue, violet spectrum here across the picture. And I want to point out that we have our own no ensemble averaging logo. If you go on to Cafe Press, you can find out about this no ensemble averaging logo. It's also on our webpage. I want to thank all of these agencies for their support of our research over the years. And you might wonder why I say this is the guacamole team. So for those of you that haven't heard this particularly bad joke, one molecule is one guacamole, right? Because it's 1 over avocado's number of moles, okay? So For that, thank you very much for your attention.